Hello students, welcome to EPZ Pathshala. I am Dr. Nirupama Trihenpati from Institute of Liver and Biliary Sciences. Today we are going to discuss the module Cytokine Therapy from paper Molecular Therapeutics. Objectives of this module and this cytokine therapy is designed to teach the students that cytokines are the messengers of the immune system. Cytokines are substances, either proteins and glycoproteins secreted by immune cells. Cytokines have role in general physiology. Cytokines have role in diseases. They have endocrine, paracrine, and autocrine functions. Cytokines have locally or at distance to enhance or suppress the immunity. I will tell you that what are the cytokines. Cytokines are a unique class of intracellular regulatory proteins that play a crucial role in initiating, maintaining, and regulating immunological homeostasis and inflammatory processes. Indeed, measurement of cytokine profiles in patients provide a useful indication of disease status. Due to their multiple functions, including regulatory and effector cellular functions in many diseases, these molecules and their receptors and their signal transduction pathways are promising candidates for therapeutic interferences. What are the cytokines? The therapeutic administration of cytokines, modulation of cytokine action, or at times gene therapy is being used for wide range of infectious and autoimmune diseases. In immunocompromised patients like AIDS and in neoplastic patients, cytokine therapies are being used. Cytokines regulate the innate immune system, natural killer cells, macrophages, and neutrophils. They also regulate the adaptive immune system, the T cells, B cells, immune responses. In the immune system, cytokines function in cascades way. Thus, clinical trials of individual cytokines are rarely useful since cytokines tend not to work individually. Some of the individual cytokines that have been tested and found ineffective for cancer treatment include interleukin-1 beta, although it may be useful because it helps to mitigate the severe toxicity of interleukin-2. Tumor necrosis factor certainly sounded promising, but in fact caused severe hypertension when used systemically. And interestingly, interleukin-4 showed minimal anti-cancer activity and was toxic also. In the same way, interleukin-6 had some activity against cancer cell but turned out to be a growth factor for myeloma cells. Granulocyte macrophage clone stimulating factor used primarily in stem cell transplant to reconstitute the myeloid series has been studied for melanoma with controversial results. What is the mechanism of cancer? In the interplay between the cancer and immune system, tumor cells employ several mechanisms to evade the immune responses. One such mechanism is the suppression of MHC class 1 molecules. Suppression of these MHC class 1 molecule prevent the immune response, which is mediated by cytotoxic T cells. Impaired MHC expression is generally common among tumor patients, and in one systemic study it was found that the majority of the MHC expression is less especially in the breast cancer samples. The immune system has evolved strategies to sense this lack of MHC class 1 expression on transformed cells. Indeed, MHC deficient tumor cells induce a strong response mediated by natural killer cells, a cytotoxic subtype of innate immune cells, and NK cells are most strong innate immune cells. If the MHC expression is less, these NK cells become energetic and then they don't perform in the tumor microenvironment. In recent publications, it was found that continuous expression, low expression of MHC class 1 make the NK cells more dysfunctional and the impaired NK cells. Despite advances in treatments like chemotherapy and radiotherapy, metastatic cancer remains a leading cause of death for cancer patients. While many chemotherapeutic agents can efficiently eliminate cancer cells, long-term protection against cancer is not achieved and many patients experience cancer recurrence. Mobilizing and stimulating the immune system against tumor cell is one of the most effective ways to protect against cancer that recur or metastasize. Activated tumor-specific cytotoxic T lymphocyte can seek out and destroy metastatic tumor cell and reduce tumor lesions. Natural killer cells are a frontline defense against drug-resistant tumors and can provide tumoricidal activity to enhance tumor immune surveillance. Cytokines like interferon gamma, anti-TNF alpha play a crucial role in creating an immunogenic microenvironment and therefore are key players in the fight against metastatic cancer. To this end, a group of anticyclines or the treatments like the photodynamic therapy exert their effect on cancer cells in a manner that activate the immune cells. Reconstitution of immune cells in cancer. Immune reconstitution is important for treating malignancies. CD4 T cells assist in recruitment of CD8 T cells and also activate macrophages through the secretion of immune regulatory cytokines including IL-2, IL-12 and interferon gamma, thereby inducing tumor cell death. This also indicates a synergistic relationship between CD4 and CD8 cells in immune responses. It was found that ratios of T lymphocyte subsets were disordered in tumor patients. The analysis showed that significantly decreased percentage of CD3 and CD4 cells and in fact the CD4 and CD8 ratios in the tumor patients. This immune function or the disabled immune function can be restored with the immune therapies. This also related with the Th1 and Th2 imbalances and Th1 cytokines including the interferon gamma, IL-2 and TNF alpha enhance the cytotoxicity of the cells, whereas the Th2 cytokines IL-6 and IL-10 are associated with the tumor immune escape. It is important that cytokine therapies increases the interferon gamma and IL-2 and maintain the homeostasis between the Th1 and the Th2 cells, which is very crucial for the cancer. Most of the cytokines used in the therapies. Diseases are treated in diverse way with the diverse arrays of cytokines. Natural cytokines, synthetic cytokines, recombinant preparation of cytokines. Most popularly used are the GM-CSF, which is called Granulocyte-monocyte stimulating factor (GCSF), interferon, IL-1, IL-2, IL-4, IL-6, IL-7, IL-10 and IL-12. With the combination of cytokines, in fact, various chemokines or the synthetic cytokine preparations were used in the patients for the cancer therapy. Other including guanosines or the other including nucleosides are being used in the clinical and the preclinical studies. Generation of tumor-specific immunity. Cytokines facilitate Th1 cell-mediated immune reactions but not Th2 cell-mediated reactions. When produced in tumors, that's the reason they are not effective for anti-tumor responses. Several technical problems to express sufficient amount of cytokines in appropriate target cell remain unresolved. But the potential of cytokine is the gene therapy. Cytokine therapy is being explored. Modulation of cancer by the cytokine and enhancement of anti-tumor responses can be induced by both single or the combination of the cytokines. Cytokines are relatively unstable in vivo, therefore it needs to administer in the large amount. However, the large amounts of the cytokines, they increase the toxicity, and to avoid these toxicity, now there is a treatment for the lymphocyte extraction. Then lymphocyte should be expanded in vitro. It should be stimulated by the appropriate amount of cytokines, and then these lymphocytes can be put into the body again and specifically target and destroy the tumor expressing antigen against the tumors which they have been raised for the cells. Cytokines are being used in tumor regressions. Individual cytokines are effective for the cancer treatment, like interleukin 1 are individually being used, interleukin 1 beta is being used. In severe toxicity of the IL-2 is observed when it is individually used. Severe toxicity of the TNF, tumor necrosis factor, is being seen when it is individually used. However, the interleukin 4 and interleukin 6, when it is being used individually, there is less toxicity. Granulocyte macrophage clonal stimulating factor GM-CSF, it is being used individually and it has the less toxicity. Cytokine treatment in tumor regression. Some of the individual cytokines that have been tested and found ineffective for the cancer treatment. Therefore, including the IL-1 beta, although it may be useful because it helps to mitigate the severe toxicity of interleukin 2. Therefore, the combination of the cytokines can be used for the tumor regression. Pro-inflammatory cytokine treatment is very important to revert the functional energy of the NK cells within MHC deficient tumors. Their combination of IL-12 and IL-18 or with the mutant form of IL-2, which is called as a superkine, can be administered in the appropriate doses. It is found that 20 to 80% improvement in the combination cytokine therapies, which is not present in the individual cytokine therapy. Now, it is important to weigh between the cytokine therapy efficacy or the versus toxicity. Toxicity also increases with the higher doses. Therefore, it should be the lower doses. Less toxic doses will suffice to prevent the NK cell energies. Not only the NK cell energies, even the MHC expression, low expression, can be rectified with the combination therapies of the cytokines. Now I will teach you about the individual cytokine and cytokine therapies. Let's talk about interferon alpha. Interferon alpha is actually a family of molecules rather than a single molecule. The interferon alpha is encoded by closely related genes on chromosome 9. It is encoding proteins that are variably glycosylated. These are comprised of about 150 amino acids and they bind to certain receptors on the surface of immune cells. Interferon alpha known to have profound and diverse effect on gene expression. on many cells and tumor environment also. Interferon alpha has many roles. It upregulates the genes like MHC class 1, tumor antigens, and adherent molecules. It is also an anti-angiogenic agent. It is extremely active in the immune system, promoting B and T cell activity. Interferon alpha can stimulate macrophages, even dendritic cells, and upregulate Fc receptors. The substance we know now as interferon was isolated from white cells and called interferon because it interfered with the viral infection, it interfered with the tumor environment, and in the tumor environment it turns out that type 1 and type 2 interferon do have some anti-tumor activity, but it is not hoped for level raised. Now I will talk about interleukin 2. Interleukin 2 is a T cell growth factor that binds to specific tripartite receptors on T cells. In dose escalation studies, patients treated with high doses of IL-2 showed clinical responses, although there was a severe toxicity. Response rates were high, 24%, and at the highest IL-2 doses in patients with the renal cell carcinoma, it shows the highest toxicity. It is a naturally produced by body T cell growth. IL-2 targets the adaptive immune cell such as T cells and B cells. IL-2 also target the antigen fighting T cells and stimulate B cells to produce more antibodies. IL-2 generates a T cell response that reacts with cancer cells and melanocytes. IL-2 in the renal cell carcinoma is most popularly used, and on the basis of duration of response, and it is the checked data whether it is toxic or not, and achievement of response was calculated in the renal cell carcinoma. Doses of IL-2 is very important to affect the concentration, and if there is no effect of concentration, inappropriate concentration will be changed by the serial doses. IL-2 also increase the lymphocyte levels and plus the initial treatment with the IL-2. Combination of interferons and interleukin 2. It is being used for the cancer therapy because it is very important to know that which cytokines are important for the cancer. It is being observed that IL-2 and interferon alpha 2b are the two cytokines approved by the FDA for treatment of cancer. IL-2 has demonstrated activity against renal cell carcinoma, lymphomas, and many other cancers. Even overall these cytokines are important to treat for the cancer patient. However, the combination therapies with the interferon and IL-2 are the most important, and it needs the less doses with the less toxicity. Anti-TNF alpha therapy. Anti-TNF alpha therapy inhibit the effect of tumor necrosis factor cytokine. It becomes important in diseases like rheumatoid arthritis, inflammatory bowel disease, cirrhosis arthritis, and ankylosing spondylitis. Anti-tumor necrosis factor agents have been the first line of treatment for moderation to severe ulcerative colitis and Crohn's disease. It inhibits the effects of tumor necrosis alpha cytokine, and this therapy was used in Crohn's disease in 1998. For ulcerative colitis, it was approved by FDA in 2005. After the approval in 2005, it has become the mainstay in the treatment. Combination therapy of anti-TNF with then IL-6 is thought to reduce the risk of immunogenicity to anti-TNF agent and potentially to reduce the disease activities. However, some patients do not respond to anti-TNF treatment or their response decreases over time. Anti-TNF therapies have been shown to trigger autoimmune responses such as lupus-like syndrome. This phenomenon highlights the need to identify new molecular targets other than anti-TNF therapy for the various diseases. As I talked about it, that anti-TNF therapy has realized us that we still need the more molecular targets, and with the discovery of anti-TNF therapy, it was thought that another molecule is TGF-beta cytokine. However, TGF-beta cytokine totally does the immune suppression. It does the immune suppression of the macrophages. It does the immune suppression for the dendritic cells, NK cells, and even the tumor cells. The targets of new therapeutic molecules in inflammatory bowel disease must aim to restore immune dysregulation by the inhibition of pro-inflammatory cytokine, which is TNF-alpha. To inhibit the TNF-alpha, there was the tendency to augment the anti-inflammatory cytokine like TGF-beta. Therefore, to pursue new anti-inflammatory targets such as the regulatory T cell therapy, the TGF-beta was the new molecule which will be used for the anti-cancer therapy. This cytokine is most potential cytokines for the cancer therapy. However, it has the dual nature. As I told you in the earlier slide, TGF-beta is the most important cytokine and it has the dual nature. It can act as the tumor suppressor to tumor supporter. Transforming growth factor beta type 1 is a ubiquitous cytokine. What it does, it has the ability to inhibit epithelial cell proliferation during the tumor progression. So, however, the somatic mutations abrogate the TGF-beta signal transduction pathway and found in many gastrointestinal cancer that its importance as a tumor suppressor. In contrast, in many non-gastrointestinal epithelial malignancies, lack of these somatic alterations and in these cancers still acquire resistance to the growth inhibitory effect of TGF-beta, where it becomes the tumor promoter and tumor supporter. In many instances, resistance is part of signaling switch whereby TGF-beta loses its growth inhibitory effects and then used by epithelial cell as the growth promoting fashion. The mechanism that underlies this change in phenotypic growth response are not being elucidated in human studies. However, it is elucidated in the mice studies. Another most important factor is the cytokine granulocyte-monocyte colony stimulating factor. This is the most important in the stem cell therapies also. With the stem cells, the transplantation, this cytokine is administered and it has given the more benefit with the stem cell therapies. This in GM-CSF, in stem cell and bone marrow transplant, it reconstitutes the myeloid series totally. The data suggests that GM-CSF administration after surgical resections of the cancers, this GM-CSF gives a significant prolonged increase in the immune responses, which that immune responses are the anti-tumor immune responses, and after the transplantations of stem cell and bone marrow, it increases the cell proliferation. Interleukin 12 is another cytokine. It is a heterodimeric protein. It promotes the NK and the T cell activity and is a growth factor for the B cells. IL-12 may have value as vaccine adjuvant. When IL-12 was paired with peptide vaccine in patients with resected stages of cancers, IL-12 appeared to boost the response of these vaccines. Along with IL-12, even GM-CSF has the same value and evaluated as adjuvant for vaccine therapy. Cytokines such as IL-2 have modest anti-tumor activity in metastasis of renal cell carcinoma and melanoma. However, the IL-12 and GM-CSF are unlikely to be stand-alone agents for any histology but show promise as adjuvant therapy and in combination with other cytokines. Association of inflammatory cytokines with cancer. In esophageal cancer, it is often observed and marked that this cancer is most aggressive and have the tumor growth and the poor prognosis in the patients. Patient group who benefit from the perioperative therapy and not yet defined the tumor microenvironment and circulating factors as possible predictor of response and prognosis gain interest. In these anti-inflammatory cytokines like the IL-6 has the strong value for the IBD as well as the esophageal cancer. IL-6 shapes the environment surrounding the tumor by directly intervening in cell growth and cell survival. High IL-6 level showed a trend to be associated with worse prognosis. IL-6 levels higher than 50 picogram per ml were significantly associated with the adverse prognosis. Along with the TNF inhibitors, we can use the IL-6 receptor inhibitors also. It binds with the IL-6 receptor and soluble IL-6 receptor and it prevents the IL-6 binding to the IL-6 receptor. TNF inhibitor and IL-6 inhibitor, they both can be used as the combination therapies for the cancer. IL-17 is also used as a therapy and it is approved from FDA. This is the first line treatment for moderate to severe plaque cirrhosis, as second line treatment for psoriatic arthritis and ankylosing spondylitis. IL-17 is a good target, especially in the psoriasis. What is the limitation of cytokine therapy? Treatment options for patients with metastatic cancers have changed drastically in these 5 years. Although the cytokine therapy increased the overall median survival of patients with advanced stage cancers, it increased from approximately 9 months to 2 years in few cancers. However, the limitation of cytokine therapies that there is a variation in dose responses. Sometimes the few patients respond with the less doses. However, the few patients do not respond with the higher doses of cytokine therapy. Cytokine therapy causes the toxicity. It increases the risk of bacterial, viral and fungal infections. And on top of that, there is a lack of clear understanding for the antagonist action of these cytokines. I summarized here, cytokines as a part of tumor microenvironment influence tumor growth and progression. The study of different cancers, it is important to note that the cytokines are individually being used and in combinations. Combination of the TNF alpha and IL-2, combination of the IL-6 and TNF alpha is also being used for the tumor therapy. However, the cytokine therapies have the toxicity problems, it has the dose response problems, so moreover the clear understanding of their mechanisms. Results are promising, but further studies, including the sequential measurements of cytokine levels, are necessary to illuminate their complex network in the tumor microenvironment. Thank you.